Imagine a bustling city, alive with activity. Cars, buses, and trucks navigate a network of roads, delivering goods and people to their destinations. Now, shrink that city down to a microscopic level, and you have a glimpse into the bustling world inside a cell. Cells, the fundamental units of life, are far from static entities. They are dynamic and highly organized, with a continuous flow of molecules moving within their boundaries. This intricate dance of molecules, known as intracellular transport, is essential for virtually every aspect of cell function. Just as a city relies on its transportation system to function, a cell depends on intracellular transport to survive and thrive. Without it, cells would be unable to deliver nutrients, remove waste, send signals, or even maintain their shape. In this journey into the cellular world, we will explore the fascinating mechanisms behind intracellular transport, uncovering the key players and their roles in this microscopic ballet of life. Much like a city relies on its network of roads, cells depend on a complex system of intracellular highways for molecular transport. These highways are called microtubules, long, rigid hollow tubes that crisscross the cell's interior, providing structural support and acting as tracks for molecular movement. Microtubules are constantly assembling and disassembling, adapting to the cell's changing needs. Imagine them as dynamic roads, appearing and disappearing as needed to direct traffic flow within the cell. These remarkable structures are composed of a protein called tubulin, which assembles into long chains, forming the walls of the microtubule. The precise arrangement of tubulin molecules gives microtubules their unique properties, enabling them to withstand the forces exerted by motor proteins and cargo. Without microtubules, the cell would be a chaotic jumble, lacking organization and the ability to efficiently transport molecules. Navigating the microtubule highways are specialized proteins known as motor proteins. These tiny molecular machines are the engines of intracellular transport, responsible for hauling cargo along the microtubule tracks. Motor proteins bind to both the cargo, such as vesicles or organelles, and the microtubules. They then use energy derived from ATP, the cell's energy currency, to walk along the microtubule, dragging their cargo behind them. There are two main types of motor proteins, kinesins and dyneins. Kinesins generally move cargo towards the plus end of microtubules, which typically corresponds to the outer edges of the cell. Dyneins, on the other hand, transport cargo towards the minus end, usually located near the cell's center. Imagine kinesins as delivery trucks, transporting goods from the cell's center to its periphery. Dyneins, conversely, act like garbage trucks, carrying waste products from the cell's outskirts towards its center for disposal. The coordinated action of kinesins and dyneins ensures a continuous flow of molecules in both directions along the microtubule highways. Motor proteins don't transport molecules directly. Instead, they rely on specialized containers called vesicles to carry their cargo. Vesicles are small, membrane-bound sacs that bud off from one organelle, carrying molecules to be delivered to another. Think of vesicles as cellular shipping containers, packaging and protecting their contents as they travel along the microtubule highways. They prevent cargo from interacting with the cytoplasm, ensuring safe and efficient delivery to the intended destination. Vesicles come in various sizes and compositions, each tailored to carry specific types of cargo. Some vesicles transport proteins, while others carry lipids, carbohydrates, or even entire organelles. The formation and transport of vesicles are tightly regulated processes, ensuring that the right cargo is delivered to the correct location at the appropriate time. Section 5. A cellular highway in action: protein transport. To illustrate the intricate dance of intracellular transport, let's follow the journey of a newly synthesized protein from its production site to its final destination. Proteins are synthesized in ribosomes, tiny molecular factories scattered throughout the cytoplasm or attached to the endoplasmic reticulum, or ER, a network of membranes extending throughout the cell. Once synthesized, proteins destined for secretion or incorporation into the cell membrane enter the ER. Inside the ER, proteins undergo folding and modifications, ensuring they are correctly shaped and functional. Next, these proteins are packaged into transport vesicles, which bud off from the ER and travel along microtubule highways towards the Golgi apparatus, another organelle involved in protein processing and sorting. Within the Golgi apparatus, proteins undergo further modifications and sorting, like attaching address labels, directing them to their final destinations. Finally, proteins are packaged into new vesicles, which bud off from the Golgi and are transported to their final destinations, such as the cell membrane, lysosomes, or other organelles. Section 6. Energy for the journey ATP. The intricate machinery of intracellular transport requires a constant supply of energy to function. This energy is provided by ATP, adenosine triphosphate, the cell's primary energy currency. Motor proteins, the engines of intracellular transport, consume ATP to fuel their movement along microtubules. Each step a motor protein takes requires the hydrolysis of one ATP molecule, releasing energy that powers the protein's movement. Think of ATP as the fuel that powers the cellular trucks, enabling them to haul cargo along the microtubule highways. Without ATP, motor proteins would grind to a halt, and intracellular transport would cease. Cells constantly produce ATP through various metabolic processes, ensuring a continuous supply of energy to power the countless reactions and processes essential for life. Section 7. When transport goes wrong. The intricate system of intracellular transport is vital for cell survival. Disruptions in this delicate balance can have severe consequences, leading to a range of diseases. Defects in motor proteins, microtubules, or vesicle formation can disrupt the flow of molecules within the cell, leading to the accumulation of toxic substances, depletion of essential nutrients, and impaired cell signaling. For example, mutations in the genes encoding kinesin proteins have been linked to neurological disorders, such as Charcot-Marie tooth disease, characterized by muscle weakness and atrophy. Similarly, defects in dynein function have been implicated in primary ciliary dyskinesia, a genetic disorder affecting the cilia, hair-like structures that line the respiratory tract and other organs, leading to respiratory problems and infertility. Understanding the molecular mechanisms underlying these diseases is crucial for developing effective treatments and therapies. Section 8. The bigger picture: intracellular transport in you. Intracellular transport is not merely a microscopic phenomenon confined to the cellular world. It plays a crucial role in maintaining the health and function of our entire body. For instance, nerve cells rely on intracellular transport to transmit signals throughout the nervous system. Motor proteins transport neurotransmitters, chemical messengers, along microtubule highways from the neuron's cell body to the synapse, the junction between two nerve cells, enabling communication between neurons. Similarly, muscle contraction, a fundamental process for movement, depends on the precise delivery of calcium ions within muscle cells. This process is mediated by specialized proteins that regulate the release and uptake of calcium ions from intracellular storage sites, ensuring proper muscle function. Disruptions in intracellular transport in these and other tissues can have far-reaching consequences, contributing to various diseases, including neurodegenerative disorders, muscle dystrophy, and immune deficiencies. Section 9. The future of cellular exploration. The study of intracellular transport is an active and rapidly evolving field, with scientists continually uncovering new insights into the molecular mechanisms governing this fundamental process. Advances in imaging technologies, such as super-resolution microscopy, allow researchers to visualize the movement of molecules within cells in real time, providing unprecedented details about the dynamics of intracellular transport. Furthermore, the development of new tools to manipulate cellular processes, such as CRISPR-Cas9 gene editing, enables scientists to precisely target and study the function of specific proteins involved in intracellular transport, paving the way for developing new therapies for diseases caused by transport defects. As we delve deeper into the intricacies of the cellular world, we gain a greater appreciation for the complexity and elegance of life at the molecular level. Understanding the mechanisms of intracellular transport is not only essential for comprehending the fundamental principles of life, but also for developing new strategies to diagnose, treat, and prevent diseases that arise from disruptions in this delicate balance.